Right. We need to talk about the forams. They actually are a group. So the next two that we talk about are groups within Sarcodina. Foraminifera is another group. It's an amoeba. It's a type of amoeba, and it moves by means of a pseudopod as well. And there's a picture of it right there on the right, and you can see that it has um, not the same kind of form that the amoeba that we think about have, but more of a star-like shape. And they have a shell, a very hard shell that's porous, and that shell is made out of calcium carbonate. Also, there are grains of sand that get caught up in the calcium carbonate. Actually, sand is a lot of calcium carbonate, but there's the calcium carbonate and other particles that cement together. And geologists have used these forams to, well, they've used the fossilized remains to determine the age of some rocks and some sediment in order to locate sites for drilling for oil. So they know that in those areas there's probably going to be oil because that's going to be the oldest part of the sediment. They do move by means of pseudopod. Next, you have your radiolarians, and they're also types of amoeba, and they have shells called tests. So the actual shell is called a test. And the difference is that their tests are made of silica, SiO3. So they are two different kinds of amoebas that actually have a shell. Now, the amoebas are going to produce asexually in favorable conditions, and they do that, and the parent cell is just going to divide into two. So it's a type of binary fission, and they divide in half, and then you have got a new amoeba. However, in harsh conditions, like you saw on the um, the diagram with the Entamoeba histolytica, there are times when conditions are harsh and they intend to form a cyst, and they will stay in that cyst for a long period of time. So if you have that particular amoeba, and there are certainly other parasitic amoebas, I know one of the more common that we're hearing about now is the brain-eating amoeba. If the conditions are favorable, you're going to be sick. But if you've got a really healthy immune system and you don't present a suitable environment for the entamoeba, then it's not going to die. It's going to just form a cyst where it can sit and remain dormant for long periods of time. And that's how, I think I've told you before, I've had a couple of students that got amoebas from eating from unclean bag salads, and it's really common to pick an amoeba up there. So watch your bag salads, even if they say pre-washed. And they both have them. One of them named hers George, and when George acts up, according to her, that means he's come out of his cyst and he's causing her to show symptoms. But when her immune system is healthy and she takes a lot of vitamins, vitamin C, by the way, is a huge help for a natural alternative to some of the um, protista medications, then George walls up in his cyst and he can stay there for long periods of time. So she knows that when she gets stressed out or when that she, her immune system isn't what it has been, or she catches a cold or something like that, this is also going to be a time when George comes back out of his cyst. It's hard, again, to get rid of them. We're talking about living organisms that live inside living organisms. So they're really, they're difficult to get rid of. So watch your bag salads. All right, winding them up, we have our phylum, which is Sarcodina. Their common names are the amoebas, foraminifera, radiolarian is in there too. I left it off. Their body plan is single-celled, although they don't all have a unified body plan. It's sort of a free-form body plan for the amoeba, the Entamoeba histolytica, whereas the forams and the radiolarians, with their shell, they have a more formed body plan. All right, they all move by means of pseudopodia, that fake foot. and their nutrition, they're heterotrophs. They're not capable of making their own photosynthesis. They don't, um, or they're not capable of making their own food through photosynthesis. They don't have chloroplast, that kind of thing. So they need to feed off of other organisms, typically bacteria or smaller protists. And of course, some of them can be parasitic. The representative genus amoeba actually is a genus, but if I ask you to give me a scientific name, of course, you could give me Entamoeba histolytica. All right, moving into Coleophora. This is our next phylum, and it contains about 7,000 species, and they're commonly known as the ciliates. If you have cilia and move by means of cilia, then that's what you're referred to. Cilia has the same protein makeup as flagella, but they're very, very short. They're hair-like projections. They extend from the cytoplasm, and they're lining the entire cell membrane, and they engulf the entire outer body of the parasite. A lot of them are parasitic, some are free living, but mostly they're found in fresh water. Now, of all the types of movement we've talked about, cilia is the quickest. And I think I've mentioned that amoeba and flagella are a little bit slower. So cilia tends to be quicker. They can beat those little cilia back and forth and move forward or backward. So this is what one on the left looks like underneath the microscope. And unfortunately, we're not in class. We would take a little trip out to the little pond in front and take a water sample, and we could see by putting several dyes on it, we could see the paramecium. We could also see some giardia. Don't know if we would see any amoeba or not, but it's possible. All right, members of the genus Paramecium, that would have to be underlined. There are tons of species of Paramecium. So sort of like Plasmodium, they just tend to lump it into a genus when they're talking about it. It is the most studied of all of the ciliates. They know more about it. They're found in ponds, slow-moving streams, any sort of stagnant water where you're going to find some decaying organic matter, decaying plants. That's where you find your paramecium. They're not going to be found in marine environments. They're strictly fresh water. All right, there's another view of what a paramecium looks like. They have their cilia arranged in rows surrounding the entire cell membrane, and the cilia beats back and forth and propels them through the water. Here's one in motion. Not very exciting to watch, but he does have um, quicker movements, and then he can be stagnant for a period of time. All right, what do they eat? They feed on bacteria, and they can feed on algae and any other of the smaller organisms that they would find in their environment. And over to the left, you can see we're gonna talk about what these structures are because they are the most commonly studied. They do have certain organelles that are special to them and definitely worth mentioning. Okay, so here's his picture over to the right, and they do have the most elaborate of organelles. They have a very clear, here's my pointer, plastic, very tough layer called a pellicle that surrounds their cell membrane. And it is got a funnel-like depression here. This is the depression, and it's called the oral groove. So with the cilia, it can beat back and forth and cause motion, but if you notice, this oral groove is also lined with cilia, so when it beats, it can bring in food into their oral groove, because if they're in stagnant water, water's not moving. They're basically filter feeders and feeding off of things that they just happen to pull in. So cilia are pretty important, not only as motion, put in feeding.